Day before yesterday, we have already completed the human genetics, and now we are going to study about populational genetics. I hope you will love this session. If you love this session, then make sure that you share this session with all your friends, relatives, and all the students you get near. So, genetics. Okay, just give me a second. Let me see whether my stream is live or not. So population genetics always starts with some genes, right? So these genes are what type of genes? The genes which is contained in a specified population or a specified area, or the genes which is there in a specified community. Then when many genes constitute a community, then these genes are known as the population that creates or makes a population. Clear? Now, today we are going to which is included in a population. Yesterday we have already studied about pedigree and the gene disorders which are present in the human. But today we are going to study about the population genetics or the genetical studies which is based on populational level. Right? Now, when we are studying about population level, then there are something which matters in the populational level. And what are those things? Let us learn. So, these things are, first is same species. Okay. Same species who are living in the same geographical area. So, same species will be living in same geographical area. This one must be same or residing in a same community, we may say, right? This is known as a population. This is known as a population. Now, what type of inheritance of characters we can see in a population? What type of inheritance of characters? So as I am teaching you, you can also write down the note, right? So what type of inheritance characters? First one is nature of gene and alleles, right? First one is genes and alleles. Now what type of gene nature will be there? The gene can be dominant, as I already taught you yesterday or in my previous live sessions, and the gene can be recessive also. So this is the nature of the gene. It can be two types. It can be dominant, it can be recessive. Next, there must be multiple allelism. The multiple allelism, what is that? I've already explained in my first live session. If you have not watched that live session, you can go and watch that live session. And in multiple allelism, I can just say you that when a single gene or a single allele, when one phenotypic effect is there, okay? When one phenotypic effect is affected by several genes or several alleles? Several genes or several alleles, right? And we get lots of phenotypes, right? So that is known as multiple alleles. That one is known as multiple alleles. Now is polygenic inheritance. Okay, what is polygenic inheritance? Polygenic means polygenes. What do you mean by the word poly? The poly means many, right? When there will be many genes and there will be only one effect. Only one effect. So that is known as polygenic inheritance. Okay. ABO blood group is an example of codominance and is also an example of multiple allelism. Why? Because here we get 4 different phenotypes, right? Here we get 4 different phenotypes. What is the 4 different phenotypes? The 4 different phenotypes is A blood group, B blood group, O blood group and AB blood, right? So you get 4 different things. And there are lots of genes also. So when there will be several genes and several phenotypes, then it will be known as multiple allelism. From the word only we can understand this. Now what is polygenic? Polygenic means many genes but only one effect will be there. There are many genes but only one effect. Like your skin color, right? That is your skin color, which is a polygenic inheritance. Many genes are there. Capital A, capital A, capital B, capital B, capital C, capital C. Or you may say capital A, small a, capital B, small b, capital C, small. Or like 7 different phenotypes are there. 7 different genotypes are there, right? And but the effect is one. Every gene, that is 3 genes, that is A, B, C, these genes are only affecting your skin color. It is not affecting other organs. It is not doing any other effect. So yes, this is known as polygenic inheritance. Next, another inheritance can be there. Now we'll study about gene frequency. That how much quantity of the gene is contained in a single population, or how much quantity of the gene participates while making a new offspring. Okay. So before studying this vivid and a word term, so what we will learn? First we will learn about genome. What is genome? So genome is the total gene content in an organism. Total gene content in an organism. As we say, diploid organism, haploid organism, polyploid organism. Okay, so it differs about our poidy level. That is, diploid are always 2n. So 2n is the total number of gene content. Haploid means n. Polyploid means more than. Many polyploidy. Many genes. So this is known as genome. Now what is gene pool? What is gene pool? Gene pool is the total gene content which is present in a specified area. Suppose this is area A, right? And this is area B. So total gene content, okay, total gene content of area A is the gene pool of area A. Is the gene pool, or the total people or the total population of area A residing contains the gene pool. There will be many population, many people residing in the same area. And if I count like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So gene pool A is containing 10 people, right? It is containing 10 person. It is a community of 10 people. And if I count like in B, okay, if there is 1, 2, 3, 4, 5, so I'll tell that in gene pool of B there are 5 person or 5 people residing in gene pool of B. So this is known as a pool. Gene which is contained in a confined area, it is known as gene pool. Now gene flow. Many a times what happens that organism tend to leave their own gene pool and visit another gene pool, right? So if the people of A visit to B, some people or relatives thought that it is Diwali, and two people thought that let us visit to gene pool B. Now there, what after Diwali, what will we see? That in gene pool A there are 8 people, whereas in gene pool B there are 7 people, right? So this is the phenomenon. This is the phenomenon. So this is the drift. This is known as when there is migration or the movement of genes. When there is movement of genes. Suppose, let me give you a realistic example. Right? You stay in Delhi. Okay? Now you thought that I'll go to Maldives. I'll go to Maldives for spending a vacation. Now some genes from Delhi, or your gene, you are carrying a gene from Delhi along with you to Maldives, right? And you're increasing the population of Maldives for a certain period of time, right? And then it is known as gene flow. When there is movement of genes from one population to another population is known as gene flow. There can be also random mating. If there is gene flow, there will be random mating. That is, the populational structure of A, the two people of A can migrate to B, or they can have random mating. So there can be migration, there can be random mating, right? There can be random mating, there can be genetic drift. What is the genetic drift? Genetic drift is just a movement of the genetical load. Okay, there was a genetical load in A, right? There was a genetical load in A population. The movement of the A population went to B, so the genetical load of A decreased where the genetical load of B increased, right? So when there will be the movement of the genetical load from population A to some other population, that is population B or anywhere else, then that will be known as genetic drift. That is, genes are drifting from its original position, right? So when there will be gene flow, there will be migration. When there will be gene flow, gene can be flow, and there will be, the cause will be random mating. Anyone can mate. The people of Delhi can mate with people of Maldives. And also when there will be gene flow, there will be movement of the genes, that is genetic drift, or the load will move. Next, now what do we mean by genetical load? Genetical load is, it is the existence of the recessive allele character in the heterozygote form in a population. So how many recessive genes are present? Recessive genes are present, like if we these type of genes we can take. This is pure dominant, right? This is pure recessive. This one is intermediate. So what is genetical load? When there will be movement of this recessive gene into the dominant, when there is suppose this is the dominant population and this is the recessive population. Now when there will movement of the dominant population towards the recessive population, then there will be random mating, and it will create an intermediate phenotype, that is capital R and small r, right? So genetical load is that only when it represents the existence of a recessive allele in the character of a heterozygote. So here it is showing the existence of small r with the capital R, right? It is showing the existence of the small r with the capital R. So this is only known as the genetical load, right? As it shows that recessive species or the recessive alleles cannot never be eliminated from a population. You cannot eliminate a recessive allele, right? You cannot eliminate a recessive allele from the population. This was the recessive population. Okay, so this B was the recessive population. So when it was recessive population, that's why it was containing less number of people. That's why it was containing less number of people. A was a dominant population, so it was containing more number of people, right? But due to migration, due to random mating, and due to genetic drift or gene flow, what do we got to see? That A and the B population got somewhat balanced, and we also got an intermediate population of capital R and small r, which is known as the genetical load, which represents the recessive allele in a population of the dominant genes, or in a population of heterozygote genes. Clear? So this is a natural population. This is a, that is capital R and small r. So when we'll get a natural population, or when we see a population which is natural, we will get both the dominant genes. Okay, we'll get both the dominant genes, plus we also will get the recessive genes. We'll get dominant plus recessive genes. There will be no population which only contains the dominant ones or only contains the recessive ones. Clear? Now we will clearly describe what is gene frequency, what the topic we have left, right? This was a topic we have left. Now we'll discuss this topic, which is very important. We need to learn what is gene frequency actually. So gene frequency is the proportion of any gene. You can also write down as I'm saying. Gene frequency is the proportion of any gene or allele of a character out of the total allele of that character in a population. Okay. So if there are a population which is containing dominant genes and recessive genes. Now dominant genes will be described as capital R, capital R. Recessive genes will be described as small r, small r. And the intermediate people or the heterozygote will be described as capital R and small r. Okay. Now what will be the gene frequency? If the total gene frequency is 100%. Okay. If the total gene frequency is 100%. Now what we can get, that this capital R type is, we may say this is 25%. Dominant is 25%. Okay. This one is 50%. And this one is again 25%. Okay. So if we say like that, then what type of gene frequency this capital R is holding, or the dominant frequency is holding? 25% by 100% equals to 1/4 gene frequency, right? What type of gene frequency this capital R and small r is holding? What type of gene frequency? That is 50%. 50% by 100% equals to 1/2 of the population. And what type of population this small r and small r is holding? This is holding a gene frequency of 25%, which is again 1/4 of the total population. So what I'd say, that gene frequency is the proportion of any gene or allele of a character out of total allele of that character in a population. Total alleles में से इसकी खुद की कैरेक्टर कितनी है, ये डिपेंड करता है जीन फ्रीक्वेंसी के ऊपर. क्लियर? फ्रीक्वेंसी को हम कैलकुलेट भी कर सकते हैं. Now how can we calculate gene frequency? How can we calculate gene frequency? We can calculate gene frequency in a given population by frequency, which is directly be calculated or indirectly be calculated on a type of character, right? In 100 human population, if I take 100 human population, now 50 population is of M type. Here we are taking an example of the Mn type of blood group. Okay. So 50% out of N type. So they will have this Mn type of blood group always has Lm and Lm, which is a pure dominant, right? Next, this is Ln and Ln, which is pure recessive, and this one Lm and Ln is codominant. Okay, that is, it is having both the characters are expressed fully in a heterozygote. Okay, both the characters are expressed fully in a heterozygote. Okay, so this is Ln, Lm and Ln. Now 50% is M type, 20 is Mn type, okay, and 30 is N type. So what I said, N will be ln ln, M will be lm lm, and this one will be lm and ln, which is a codominant, right? Now we can find frequency of M allele. We'll find, how we will find, 50 into 2 plus 20 by 100 into 2, which is almost 120 by 200 equals to 0.6. That is 60% is of dominant character. Clear? Next, frequency of M, similarly in allele frequency, that is 30 into 2 plus 20 by 100 into 2 equals to 30 by 200 equals to 0.4, that is 40%. Next, so what we got the total frequency? Sorry. So what we got the total frequency? Yes, it is 0.6 plus 0.4 equals to 1. That is 100%. Okay, that is 100%. So this was all about the gene frequency, or to how to calculate gene frequency. Gene frequency will be, that is, we can directly calculate the frequency when we can make out the genotypic from the phenotypic. That is, if there is such phenomenon, when we can directly calculate the gene frequency. Let's see, when we can directly calculate, direct calculation is there when there is phenomenon like codominance. When there is phenomenon like codominance, like this was the phenomenon, or this was a codominance example. When there is incomplete dominance, then also we can calculate this, when there is incomplete dominance. When there is polygenic inheritance, like your skin color inheritance, so when there is polygenic inheritance, we can calculate this directly. Okay, but indirect calculation, what is the indirect calculation of gene frequency? What is the indirect calculation? So indirect calculation is your Hardy-Weinberg principle. Okay, Hardy-Weinberg also gave this law, that is 0.6 in any random mating population which is non-directional, where there will be no mutation, etc., etc. Then there will be this will be the gene frequency, that overall gene will be same, that is 100%, 0.6 and 0.4 will be 1. That is, overall gene frequency will be 100%. And among these, M type is having, that is dominant one will have 0.6, whereas the recessive one will have 0.4. Clear? So this was a Hardy-Weinberg principle, or Hardy-Weinberg hypothesis we may say, or Hardy-Weinberg equilibrium, or Hardy-Weinberg law, that whatever be the gene, whatever be the migration, emigration, death or birth of an organism, every time the genetic frequency or the gene frequency of a given population will be constant, that is 1 or 100%. Every time it will be a whole 100%. Clear? So we have already discussed about this. Now we will move to the next, that is frequency of the population always remains constant. In a large randomly mating population, the frequency of any of the gene or the allele remains constant for generation after generation. If there will be generation gap, there will be more births, more dates, but also the frequency will always remain constant, right? And in Hardy-Weinberg principle, what were the factors that were not included? That selection, the nature selects the one who is adapted. So in selection, mutation, there can be mutation, there can be different allele from the dominant and the recessive, right? This was not included. Migration was not included in Hardy-Weinberg principle. There was no migration, only there it was a large closed population, where you will get the mating or the maters in your own population only. You don't have to find and find the maters in the other population. You can find the mates in your own population. You will mate with the your same population mates, and there will be random mating among your mates only. Okay. Next, population is very stable. There is no movement of the genes, there's no movement of the gene flow, genetic drift, etc, etc. There's nothing in the Hardy-Weinberg hypothesis. The population is very stable. But Hardy-Weinberg principle or Hardy-Weinberg population, this population has no real existence, because there's no such population which is infinitely large, and there's no such population where selection, mutation and migration is not going on, and there's no such population which is stable. Every time death, immigration, migration, birth and natality, mortality, each and everything, selection, mutation, etc, etc, and most probably recombination is also going on. Okay. Now coming to the frequency. So according to Hardy-Weinberg principle, what do we got to learn? According to this Hardy-Weinberg principle, so according to this Hardy-Weinberg principle, we got to learn that frequency of the dominant allele is frequency, this P is denoted as the frequency of dominant allele. Okay. And this Q was denoted as frequency of a recessive allele. And as we said that 0.6 plus 0.4 equals to 1, so P plus Q equals to 1, which is the frequency of allele. Frequency of allele. This was calculated in frequency of allele. This is totally indirect calculation. This has no real existence, but it matches with the real existence. That's why Hardy-Weinberg principle hypothesis has got place in our study. Okay. Now what we can calculate, that is P squared plus Q squared equals to, plus 2PQ equals to 1, right? Which is this one, that is P square. What is this P square? I'll clearly denote. This is actually the frequency of homozygous dominant organism. This one is frequency of yes, it will again be homozygous recessive organism, and this one is the frequency of heterozygous organism. Okay. Now we will do a numerical, because in genetics numerical are very important. So we'll do a genetics numerical. As we are studying about population in genetics, we will clearly study about detailed population, how the population is formed, and we'll do some numerical also. So your first question is, if the frequency of the recessive allele of a character is 0.3, so recessive allele का जो character है उसकी frequency आपको दिया है 0.3, because Q was the recessive allele, we know, right? We'll follow again P plus Q equals to 1, this formula. This formula will follow, and Q is 0.3. Now you have to find what? You have to find number 1, that is homozygous dominant. We have to find homozygous dominant ka frequency, right? Next we have to find heterozygous dominant ka frequency, and third one we can find that homozygous recessive ka frequency. This is hetero. Okay, this is homo. This one is heterozygous dominant. I'll write again. So take out your pen and paper and start doing this sum. Okay. So here we can get that P equals to 1 minus Q. Okay. So what is 1 minus Q? That is 1 minus 0.3. So what we will get? We will get 0.7. Okay. 0.7. So P is 0.7. Okay. Now according to our new rule, what we will do, we will take out P square, that is 0.7 whole square. So what we will get? 0.5. 0.49, that is 49%, which is relevant to 49%. Next, q square equals to, we will get q square, 0.3 is whole square, so what we get, 0.09, which is equivalent to 9%. Okay, now left is 2pq. So what is 2pq? 2pq is 2 into 0.7 into 0.3, which is 0.42, which is equivalent to 42%. Right? Now we'll do a verification that whether we have done this is correct or not. So to verify, verification is going on, and to verify we'll see 49 plus 42 plus 9% equals to 100%. So the gene frequency is 1. So we have proved this one actually. We have proved the Hardy-Weinberg principle, that is p plus q whole square equals to always 1, or p square plus q square plus 2pq equals to 1. Now it proposes Hardy-Weinberg principle. So this was problem number 1. Similar problems we'll get. Let's move on to problem number 2. Okay. Let's move on to problem number 2. If 80% allele of a character are recessive. Okay. If 80% allele of a character are recessive in a population, okay, then find out the percentage of, so what is the percentage we have to find out? Number 1 is organism with dominant character. Okay, and number 2 we have to find out the homozygous organism. How many homozygous are there? So this is the population. I hope you are able to understand. Now we'll have this. Okay. So what we get, that q equals to 80%. Right? In the previous slide, what we got, that 0.49 equals to 49%. Right? Now 80% will be equal to 0.8. Right? 0.80, or if we can neglect the 0, then it will be 0.8. So here also 80% equals to 0.8. Clear? If 80% is 0.8, then what will be p? P equals to 1 minus q. So 1 minus 0.8 equals to 0.2, which is equivalent to 20%. Right? Now, number 1, dominant character. Now what is the dominant character? Dominant character was p. According to Hardy-Weinberg principle, dominant was p, right? So the dominant character, will find out this dominant character, that is therefore p square plus 2pq equals to what? 0.2 plus 2 into 0.2 into 0.8. So what do we get? 0.04 plus 0.32, which is equivalent to 0.36. Yes. So this was the dominant character. That is 36% is dominant. Next is, they're talking about homozygous organism. Right? So what they're talking? They're talking about homozygous organism. Now how we will find this homozygous organism? This was number 1. This was number 1. Now show you number 2. How will you find out this homozygous organism? We'll find out homozygous organism by p square plus q square equals to 0.2 whole square plus 0.8 whole square equals to 0.04 plus 0.64, which is almost equals to 0.68, almost equal to 68%. Okay, so one is 36%, one is 68%, and this was only the answer. Now after this I give you a sum, which I'll not do, but you will do as your homework. So sum number 3, if the frequency, after doing this sum, if you are able to complete this sum clearly, then you will say that yes ma'am, I have understood. If the frequency of recessive phenotype in a population is 0.09, then what percentage of organisms are heterozygous in this population? Okay, just you have to find out the heterozygous. This is very easy, right? I'll also give you a hint. Well, so what I have told, that this is homozygous dominant, this is homozygous recessive, and this one is heterozygous, right? Q is already given. You have to find out the P, and then you have to find out the total heterozygous population. This one, nothing else. Now we'll study about factors which were affecting Hardy-Weinberg principle. Okay, so factors which were affecting Hardy-Weinberg is, first, due to this factors only, Hardy-Weinberg principle is not every time well accepted, and it is not considered as a law, but it is considered as a hypothesis. Okay, which is somewhat correct, but every time it is not correct according to the real existence. Hypothesis means which does not have a real existence. So this does not have a real existence. Hardy-Weinberg principle does not have a real existence, because there will be no infinitely large species where you will get all the matters of your choice, right? So if you want the matters of your choice, you have to move outside of the population. So first is selection. So Hardy-Weinberg didn't told that any people or the nature will select the individual person. Hardy-Weinberg has not told about that. Next is mutation. Okay, Hardy-Weinberg principle was not based on mutation. Next is migration. Next is genetic or random drift, that is movement of the population, right? If we draw like this, if we draw a bottle, which is known as bottleneck effect, right? We'll study about this also in this class. So if we study bottleneck effect, so you can see that this one has a large space, right? And this represents a large population. But gradually, as we are coming to the end, we can see that this is very narrow, right? Very thin and very less. So narrow, thin and less population, or less density we can find at the neck region. Why? Why we can find less density at the neck region? Because at a time there was a lot of people, lot of population, lot of person, lot of organism when it was a when it was a closed population. Now, due to migration, due to selection, due to genetic drift, and most probably due to the gene flow, gene has been flown from this position to this position, and many small population has been grown up from this large population. This was once a large population, and gene has flowed in many several ways, and the gene has flown out. So people have made their own families. This is family 1, this is family 2, family 3, and each and every is constituting a small population. Each and every family is constituting a small population. This one is another small, this one has another small population, this one is another small population. Okay, this one is another small population. So everyone is constituting a small population. And what happened to this population? What this was a parental population. What happened to the parental population? The phenomenon what happened to the parental population is known as bottleneck effect, and the parental population got decreased day by day, and ultimately there were few members left. There were only few members left. Okay, so when there was few members left, then it is known as gene flow or genetic drift or random drift. Clear? Now, migration do not affect the frequency at large scale. If there is migration, if suppose there are total 1 lakh people residing in a place which is named as place Z. Okay. Now from place Z, if 2 people goes to visit place B, right? If 2 people goes to visit place B, will it affect from the 1 lakh population? Will it affect? No, it will not affect. It will not have any effect. Migration will not have any effect in large population, right? But if there is 5 people living in a building, suppose the block, living in block C, right? And these people thought that this building is a horror place, right? This building is a horror place, and they thought that we'll move from this horror place to another block. So they moved to block number F, right? So when people or when 3 people or suppose 1 people only move from block number A, then how many people will reside? We can count in finger. So the people which are left over in this horror block are almost as finger counting people. So very, very less quantity, very, very less in number. So when there will be small population, then it will have a large effect of migration. Migration will have a large effect in small population. The people, the cheetahs or the tigers are being destroyed in the wild, right? That's why the tigers or the cheetahs are, or the elephants or the rhinoceros are destroyed in the wild. That's why the rhinoceros are protected in the national parks, wildlife sanctuaries, just to be protected, to be extinct from the wild. If they will be destroyed in the wild and they will be extinct in the wild and their survival will decrease day by day, then there will be a very small population, right? And among the 3 rhinos, if there are 3 rhinos in a single national park and 1 rhino is dead, then it will affect a great it will have a great effect on the population. The 2 rhino who are still alive will face a verge of extinction. Their category will be very, very diminished, and one day they will be totally destroyed. Clear? Now, migration, but there is, when there will be any certain change in the gene frequency, then it will be known as genetic drift or genetic drift. Okay, so that will be known as genetic drift or gene flow. Sudden change, matlab if there will be your such as there will be any reason due to coronavirus. What happened that many people who were residing outside India, they all moved back to their own country, right? They all moved back to India. So there was a genetic drift, and there's a gene flow. There was a random movement of the people, and which led to the change in the population of India also, and also change in the population of the outside area where the people were residing. Clear? So this we can we can also sample some error in cases of genetic random drift. There can be error in counting. Suppose people are moving during the coronavirus, so there will be a very big difference between the population before coronavirus in a particular area and after coronavirus in a particular area. It will also create a error in the census, because when there will be vote or election, the people there will be census, the population will be the population of the area will be counted, and after coronavirus it will be seen that the population is increased in a drastic number. So where were these people? These people were residing in some other area who have came to our country again, back to our country. Okay, now we will study about gene expression in a DNA, okay, which is very important. Okay, so we'll study about gene expression. So how is gene expressed, or what is gene expression actually? So gene expression is the second most important function of the genetic material, right? It is the second most important function of the genetic material. First most important function of genetic material is inheritance, and the second most important function of the genetic material or the DNA is expression. How will it express itself? So how it expresses? It expresses itself in ways which has been described by many of the scientists. Okay, first function is transfer of to the next generation. So you please do write it down. So first one is inheritance. Excuse me. Second one is expression. Okay, so these are the most important function of the DNA or the genetic material These are the two important function of the DNA. So actually what happens, it is the molecular mechanism. Okay, it is a molecular mechanism by which the hereditary information present in the gene comes out and produces a character. Okay, so this, which was after inheritance, what happened, that some hereditary information was contained in this inheritance, right? There was some information was there present. There was some information was present in this when there was inheritance, when the transfer of the character, there was some information was present. Now it's time to express this information. Now it's time to show this information. When there will be show of the information, that is known as your gene expression, or gene expression can also be termed as a molecular mechanism. Okay, it is termed as a molecular mechanism. Clear? Now, there are many scientists who studied gene expression and gave us the law. So first one is A. Garrett. Right? Yesterday I told you who is A. Garrett. A. Garrett is the father of modern genetics. I already told you. So A. Garrett gave a hypothesis that is one mutant gene equals to one metabolic block. Now, what do you understand by the word metabolic? What do you understand by the term metabolic, right? What do we understand? Yes, we understand that metabolic is a function. Metabolic block, or we may say a functional block, right? At a functional block. So a mutant, if there will be a mutant gene, then will be a functional block in our body, right? Metabolism is always considered a function in our body. Next, two more scientists came, very famous scientists named as Biddle and Tatum. Okay, named as Biddle and Tatum. Now, what does the Biddle and Tatum did? So, Biddle and Tatum gave a very, very, very important hypothesis, which is very, very used. That is one gene, one enzyme hypothesis. Okay. So, one gene, one enzyme hypothesis was given by Biddle and Tatum, which is very, very helpful nowadays. Why? Because Biddle and Tatum worked on whom? The Biddle and Tatum worked on the neurospora. Where did they work? They worked on neurospora. Okay, which is the Drosophila of the plant kingdom. Which is the Drosophila. In our animal kingdom, we just torture Drosophila, right? We just do every experiment on the Drosophila. So similarly, in our plant kingdom, we do every experiment on neurospora, which can grow easily due in a minimal medium. If we give a minimal tissue medium, right? Tissue culture, we do tissue culture. So if we give a minimal tissue culture medium to this neurospora, okay, if we give a minimal, let me write down minimal medium. If we get, so what is this minimal medium? This is just a sugar. We give sugar, we give salt, okay, we give biotin. Along with this, we give ammonia, which is a complementary product. If you want, you can give ammonia, or else you can just or else you cannot add it. It's a complementary product. So yes, so a neurospora, when a neurospora can grow, it is a wild type of neurospora which can grow easily on the minimal type of medium. Okay, they can grow easily on the minimal type of medium. Now, what happened? What type of experiment does Biddle and Tatum do? We have to learn about this experiment. What type of experiment does Biddle and Tatum do? Let me take more slides. So I'll make a PDF of these slides and I'll put it in the description. Now, coming to Prototrope. Well, So what is a prototrope actually? Prototrope is nothing but a wild neurospora. Prototrope is nothing but a wild neurospora. Okay, it is a wild neurospora, wild type neurospora, because many organisms are there which has two type of genes, like the one is the wild type gene, another is your recessive gene. Okay, the wild type or the dominant gene is known as the wild type gene. So wild type neurospora is a prototype. So one test tube was taken like this, right, and neurospora was taken here, and this neurospora in the test tube was exposed to what? It was exposed to X-rays. Exposed to X-rays. Okay. Now, what happened? When it was exposed to X-rays, it induced mutation, which induces mutation. So there was mutation in this wild type gene, okay, wild type gene of neurospora, and they obtained what? Biddle and Tatum, while doing this experiment, they obtained many mutant forms which are now unable to grow in minimal medium. Okay, they obtained many mutant form which are unable to grow in minimal medium, right? But the wild type neurospora was able to grow in the minimal medium. But now this neurospora, this mutant neurospora, where the mutation has gone, where they have changed their genetic makeup, right? Then when they have changed their genetic makeup, so they cannot grow in this minimal medium, which was a sugar salt solution with ammonia, right, and biotin. They cannot grow. So what happened? They will grow when some addition of nutrients are added. So what happened? Yes. So what happened? They were growing, or they will grow when there is addition of nutrients. When there is addition of the nutrients, some nutrients. So what is the addition of the nutrient in the medium? So they added such nutrient is known as autotrope. So what they added? They added autotrope in this. First there was prototrope, right? Then it was exposed to X-ray. It induced mutation. They obtained many mutant form, right? They obtained many. What they obtained? They obtained many mutant form, and this mutant form could not grow in the minimal medium. They need some extra nutrients, and this extra nutrients was known as, in the form of autotrope, was added, right? It is a mutant type of neurospora. Some extra nutrients, such nutritional mutant is known as autotrope. So first it was prototrope and then it was changed to autotrope. Okay, first it was prototrope and then it was changed to autotrope. Now what is autotrope? Autotrope is a mutant of neurospora. Autotrope is the mutant of neurospora. Clear? Now after this, they made several crosses. They selected, and 3 types of autotropes they found with different chemicals. First autotrope was with ornithine, citrulline, arginine chemical medium, which was getting enzyme A. Next autotrope, they got 3 main crosses. Okay, I'll show you. I'll not explain in detail, but yes, obviously I'll clear 3 type of why this Biddle and Tatum gave 3 enzyme, that is 1 enzyme, 1 autotrope. Okay, so first they got class 1, class 2 and class 3 type of autotrope. They got 3 types: type 1, type 2 and type 3. Okay, so this type 1 was containing enzyme. They got some enzyme, different enzyme. That's why it was totally different from type 2, and it was known as enzyme type A. Okay, this enzyme was known as type A enzyme. Next, type 2 got another enzyme which was they can grow, either there will be C2-line or there will be arginine in the medium. So they get the enzyme which is muted. They get enzyme that is a muted enzyme. So they get B muted enzyme. They get the enzyme which is B muted. Okay, is this clear? B muted. This they get A muted. Some are different enzyme which was named as ABC. And the third type got the C mutation, different mutation from the first two. So this one known as C muted, right? And what was the raw material provided to these classes? The raw material is ornithine. Okay, the raw material is citrulline, and the raw material is arginine. Okay, this one was first class for class 1, this one for class 2, and this one was for class 3. Now this ornithine got gene A, citrulline got gene B, and arginine got gene C. Now I hope it is clear. Thus they concluded that Weddell and Tatum concluded from the biosynthesis pathway in the neurospora, they concluded that each gene forms a special enzyme. What they concluded? They concluded that each gene, so conclusion of this total experiment by Weddell and Tatum was each gene forms a special enzyme. Clear? So after this we got that one gene, one enzyme hypothesis. Okay. Now this one gene, one enzyme hypothesis, this was a hypothesis, one gene, one enzyme. Okay. It was given by whom? It was given by Weddell and Tatum, who worked on neurospora, or you may say wild type prototype neurospora, from where they got autotrope, right? From this there was a modification. There was a modification by some other scientist who is named as Janovsky. Janovsky introduced a modification of this thing, which came to known as one gene equals to one polypeptide, right? One gene equals to one polypeptide. So from this again we got a new theory which said that one cistrone equals to one polypeptide. Okay. What is a difference between cistrone and a gene? What is the difference between cistrone and a gene? Cistrone is the smallest part of a gene. If we break a gene into pieces, then what we will get? We will get cistrones. So one polypeptide hypothesis came into work. Okay. Now according to this, we got, now this was the total conclusion of Weddell and Tatum. Okay. Tomorrow in the live session at 12 p.m. we will start with types of genes, right? There are so many types of genes, I'll say you, like overlapping genes, non-consecutive genes, homoeotic genes, transposons, pseudoalleles, and these are very, very important. And along with types of genes, we will also complete regulation of gene expression. Okay. Today, we started with populational genetics, and we completed what? We completed the gene expression. Up till gene expression. What is gene expression and how it was done? And how from one gene we get one polypeptide hypothesis. We completed up till this. Next, tomorrow at same time at 12 p.m. we will have types of genes and a regulation of gene expression. So if you love this video, make sure that you follow my channel and also subscribe my channel to never miss an update from my channel when I'm live, and also turn on the bell icon. So I'll show you all the overall note. You can, this one, from here we started actually. Then we move on to the populational level. We studied about gene frequency and each and everything. Next, we studied about how to calculate gene frequency in 100 human, if there is 100% or 100 human present, right? Then we studied about Hardy-Weinberg principle, which is actually a hypothesis, right, in a population which doesn't have a real existence. Then we calculated how Hardy-Weinberg genetical numerical calculation we have done. We have done 3 numericals, and this was considered as a homework given to you all. So I hope that you can obviously do this homework and let me know the answer in the comment section or the chat section. I'll verify it. If you're right, you will get a love react from my channel. Then is gene expression, that is molecular mechanism, and it is a molecular mechanism. We studied about neurospora, one gene, one hypothesis, autotrope, etc., etc. Next, we studied about, next we have completed this, and tomorrow we'll study about regulation